All right. Thank you guys and welcome and thank you for joining the very first biogeography seminar of 2026. So today we're going to have Dr. Pedro Peres-Neto talking to us, but before we do that, I do have a couple of announcements for you. Sorry. So first off, if you haven't heard already, we do have a location picked for TIPS 2028 and it will be Beijing. Now 2028 is a long ways away, but it'll be here before you know it, so keep that in mind. The other thing is that you should keep an eye on your inbox. So we are going to be working this year to bring back the Tips newsletter. So that'll just be a once a month convenient email that takes everything Tips related and puts into one place for you. So that'll be a place to get up to date on Tips events, workshops, conferences, things like that. Place to put a spotlight on some of our members or talk about publications just released in our journal, Frontiers of Biogeography. Place to put maybe hiring opportunities if you're looking for postdocs, grad students, or more. And possibly other things. If you have suggestions or things you want to see, we are always very happy to hear your feedback. So that's all I have for announcements. As always, the way this is structured, it's 45 minutes for a talk, 15 minutes for Q&A. If you have general comments, chat, things that doesn't need to be read out to the whole audience, you can put that in the chat function. When you ask questions, you have two options. If you want to ask the question yourself, you can press the raise hand button during the Q&A session. We'll elevate you and have you speak the question yourself. Or if you prefer to type it into the Q&A box, you can do that at any time and we'll read it out on your behalf. And looking forward to next month, our seminar speaker for February is going to be Juan Pablo Quimbayo, currently an assistant professor at the University of Miami in Florida. That'll be February 25th. The time of the actual seminar and the details will be out shortly, so keep an eye on that too. And always, thank you so much for attending. This will be available on our YouTube channel at International Biogeography if you want to see this again in the future, or if you maybe are from the future and weren't able to attend, in which case, hello. So with all that said, I will pass it over to Marta to introduce our speaker. Thank you.
Thank you very much, Hikaru. Yes, welcome everyone to our very first Funk Seminar lecture of 2026. We have an amazing lineup of speakers for you over the next several months, so please stay tuned for future announcements as well, and please keep coming back. We love having everyone here. My name is Marta Jarzena, and I'm the Vice President of Public Affairs and Communications at the International Biogeography Society, in case you haven't yet seen me or met me. And we are very, very excited to start this year with a great speaker. And I am pleased to introduce Dr. Pedro Peres-Neto as today's seminar speaker. Dr. Peres-Neto is a professor of biology at Concordia University, and he holds the very prestigious Canada Research Chair in Spatial Ecology and Biodiversity. He is a lifetime fellow of the Ecological Society of America and currently serves as editor-in-chief of OICOS. He previously held leadership roles as vice president of the International Biogeography Society and the editorial board member of Ecography, Global Ecology and Biogeography, and Methods in Ecology and Evolution, and of course, Frontiers of Biogeography, so our journal. In his research, Dr. Peres-Neto integrates ecological theory with rigorous quantitative methods to yield critical insights into predicting community dynamics and biodiversity change across ecological and evolutionary scales, from local to global ecosystems. And today he will talk about some of his work, focusing on ecology and evolution in spatially structured environments. So without further ado, I turn it over to Dr. Peres-Neto.
Sure.
Pedro, you're welcome to share your slides now.
Yep. Thank you. Well, thanks, Hikaru and Martha and Tibbs for organizing this series. It's fantastic. Always catch up. Previous talks, I think, is a wonderful way to continue to connect people throughout the year. Before I begin, I want to take a moment to remember Gabriel Qatar, a good friend, collaborator, who passed away this past January 18th. Gabriel brought a rare mix of curiosity, generosity, and clarity to many aspects of the work that I'm going to talk to you. We've been collaborating for a few years, and I would invite you, to those wishing to explore his work, to look into his Google Scholar profile. So with that, I want to talk a little bit about what I'm going to try to speak to you today. I divided the talking for more or less chunks. We're going to talk about the statistical moments of environmental spatial structure, how I think about them, how the spatial structure environment shapes ecological organization. I also want to make a point why I think that the spatial structure of environments themselves haven't been often, they have been often in some ways neglected in ecological research. And I want to show you a few what I call vignettes illustrating some of the ideas that I've been thinking in terms of how to integrate the structure of environments themselves into ecological organization. So obviously we all know that spatial heterogeneity is a constant theme in ecology, from their environments to other aspects of populations, communities, and ecosystems. And there has been a huge amount of decades and decades of work trying to understand this variation in space. So one of the things I like to start is the lingo of spatial structure. I think sometimes we use some of this terminology in some ways loosely in terms or sometimes different from each other. I don't want to tell you how you think about these things. I just want to tell you more or less the way I want to organize these aspects. And sometimes I'm using a little bit of text. I find sometimes watching talks, particularly if I'm not engaging, participating, helps me to have a little more text so that I can revisit those concepts. So online talks, I like to make use of them. So spatial heterogeneity is one of those concepts that you basically think in terms of how much variation and you have in space. Spatial structure and scales, those are a little more sometimes tricky to have a consensus. And so I will define for the time being, but then a little more structure that spatial structures observed in the form of distance-dependent similarity, autocorrelation, and hierarchical multiple scale organization nestedness. As ecologists, we're fascinated by these two aspects, and I feel that sometimes they're treated in some ways differently. And I'm referring here to spatial connectivity, or is the extent to which environmentally structured landscapes create predictable pathways of dispersal. So you can see here if an individual is here to disperse here, is much easier to predict than in other parts of the landscape, just by the way the spatial structure of the environment is organized. And I refer that to spatial environmental connectivity or spatial connectivity. So the moments, those are statistical moments that I like to clarify them because I'm going to be using quite a bit of that during the presentation today. So we could basically think about this as a quadrat in space, and we could take the average here of, say, environmental features or climate features here and here, and the difference that we find between those two is what I'm calling variation in means. And then we could have variation in means across nested scales. So you could have the mean here is different from this mean here. And so obviously this mean here will be different from this mean different from this. So this idea of nested scales and ecological structure is a common theme that we find in many aspects from behavior, population, community, and ecosystems. Then we obviously have also variation in means within not only between, but also within. So we can think that the average here is different from this one, and so on. So there's this other aspect of spatial heterogeneity. And the final moment, at least for today, is the spatial covariance or autocorrelation, in which we can have very random environmental features in the landscape. Those are rare to be observed. Nature tends to be quite spatially structured, but from weak to intermediate to very strong spatial autocorrelation, or often in statistics we refer to spatial covariance. One of the themes that I find in the literature and sometimes in my own work is this distinction between autocorrelation and autocorrelated and nested scales. Some ecologists will be very interested in nested scales, others in autocorrelated. Rarely, actually, I see those two types of scales being integrated in work. So I want to invite people to start thinking about how to perform this integration. Sometimes easily said than done, but I try here a cartoonistic representation of how those two can be in some ways merged, and by no means they are mutually exclusive. And there will be in depending where this variation is happening, stronger or weaker. So this is some of the work that we started doing this past year. I'm not going to speak so much about this, but this sort of helps to organize a few of these ideas. So you could have the total environmental heterogeneity in space, and this can be decomposed in different scales. So for instance, here is just 3 scales, but you can think of as many scales as possible. And we could partition this into a mean effect, a variance, spatial variance, or spatially structured variation or autocorrelation, and then here a non-spatial within site variation. So have non-spatial, spatially structured, and variation in that variation in means across space. And you can see that this can vary within a particular scale, and then we can go through sites within regions and then among regions. But obviously, you can derive this. And in the future, what we've been working on is to try to actually decompose the contributions of each of those moments to a particular, say, site, patch, or geographic self, or a lot of biogeography is done at that level, and to see how is that contribution can, for instance, explain species distributions or species richness and other aspects of ecological variation. So today I want to make this link that I've been working quite a bit on this. Others have been working on that too. Is this idea that the spatial structure of the environment itself can mediate species interactions, dispersal, and other aspects, life history, and so on, that are important for phenotypes, meta populations, local communities, meta communities, biomes, and ecoregions. So the idea here is that this changes in scale and the changes in heterogeneity in spatially structured and non-spatially structured can affect. So I'm going to try to convince you today with a few examples of how this can happen. So community ecology, it's or biogeography. Sometimes, I love the biogeography meetings, and I feel that a little bit out of sorts sometimes, because I consider myself often as a community ecologist. But then when I go to work with community ecologists, I keep thinking about large scales and biogeographic patterns and processes and so on. So this idea here is that the units that we are often interested is a site by species. I reverse this so that the matrix, usually we go sites by species, not species by sites, but this makes those matrices to be compatible in their dimensions. But the idea here is that we've been exploring ecology, a lot of complex interactions between traits and environment, biogeography and trait interactions. Talk to you a little bit today about spatial interactions, spatial phylogeny. So there's all this huge amount of work that ecologists have been put together to understand the variation. And I think that these complex interactions, they often take place they're mediated by the spatial structure of the environment in itself, as I'm going to try to demonstrate to you today. Community ecologists are fascinated by this expression that John Lawton published about 27 years ago, because where he sort of attained that community colleges often see as a mass because of seemingly vast array of processes that can underlie ecological patterns and apparent uniqueness of each study system. I think ecologists now are aware that we have to embrace complexity more than ever. And Gabriel Kattar's work definitely contributed quite a bit for us to understand in the way that those interactions or contingencies they occur to form and organize meta communities. So the idea here of spatial contingency that sometimes I use is I'll define as ecological patterns and processes that vary across landscapes because different in spatial configurations or autocorrelation or nested scales in environmental heterogeneity lead to different outcomes. So you could think that you could have exactly the same regional species species pool, and depending on if the species is in this landscape or this landscape or this landscape or this landscape, and actually I made a point here to have actually the variance to be the same. Everything that is varying here is just the spatial covariance or spatial autocorrelation. And this could lead to very different ways in which those species sort themselves into space. So this is not new. Ecologists have been very much interested by this idea of how heterogeneity, in this case, is variance, not so much the spatial covariance, but this is the total variation that we have in space. And this can lead sometimes to different types of species that are selected. And for instance, a more heterogeneous environment could select for more generalist species, whether in other cases for specialists, species are not their selection is decreased as you increase high heterogeneity of environment. So this will depend often from the types of species that you have in a pool. But what I'm trying to portray here is this idea that we could have two types of relationships here, and this is a form of spatial contingency because those relationships themselves, they change. So I'll present a few of this work today. This work comes from collaboration within the lab, different versions of it. So we demonstrating here that heterogeneity. So you could think about this is the US that we sort of did some work using the angiosperm distribution. And what we're interested is we divided into 50 by 50 kilometer geographic cells, and within each of them 1 by 1 kilometer. And we calculated what is the heterogeneity, the variance, only the variance, not the spatial covariance, although we have other work doing that, but just the variance within each of those 50 by 50 cells. And you can see that the mean niche breadth of species in terms of climate preferences, the standard deviation of niche breadth, meaning some of those cells have more specialists and generalists, so that variation, species richness and functional diversity. And as you can see, sorry about that, as you can see, that variance within 50 by 50 kilometer scale is a very important predictor for those different ecological components. So just the heterogeneity itself can be a strong driver of different aspects of biodiversity. This is work here from in Canada, in which those are watersheds. Each of those watersheds here in different colors, they have a large number of lakes. Those are about 10,000 lakes. And as we can see, when we go to the northern lakes, the environment is much more heterogeneous, is much smaller compared to the southern parts of Ontario. So that heterogeneity is reduced in the way biogeographic patterns, the outcomes of biogeographical patterns are very related to that heterogeneous variation that we find. So in northern lakes where the environmental variation is reduced, we find more nested watersheds. Each of them tend to be like this. And as you go south, the species tend to sort themselves. And so this is important because of species interactions, where here you have stronger species interaction within each of those blocks of cohesive co-distribution, whereas here you have different types of potential species distributions. So the spatial heterogeneity itself can drive the types of pools that you expect to more strongly interact within those meta communities. And that's a lot of work that Gabriel has done in the past 6 years. And so, again, I want to spend a little time on this component here, the spatial autocorrelation or the spatial covariance. One of the things that we find, and I guess I did quite a bit of work in the tools that ecologists use to understand the contributions of environmental variation, spatial variation, and so exercise as variation partitioning. And what we're going to often find is that this fraction here, we often call the B fraction, which is the spatialized environmental heterogeneity. We often discard that part. Some ecologists would treat that as a nuisance. So we can't separate whether it's environment or we can't separate whether it's space. So this is the pure environmental component, independent of space. So ecologists hope that by being pure, no spatial variation, you can use that to generalize better across different scales because they're independent of space. Whereas here, the spatial component here, some could interpret as dispersal, others could as missing predictors, and there's a long and rich literature with these debates. But what I'm trying to tell you here is that the spatialized environmental heterogeneity, this fraction here, where environment is autocorrelated, that in itself has been a place of little attention, I find, by ecologists. So I want to bring to you the importance of that. And so there are thousands and thousands of papers speaking about the issues of spatial autocorrelation when modeling ecological patterns. Species distribution modeling certainly has a vast literature in that. My point is that we pay a lot of attention in that space and maybe less attention to the fact that the environment heterogeneity itself has a spatial structure that is important for ecological interactions. So the fear of the spatial autocorrelation is more or less this cartoonistic, very simplistic representation here, in which when we know that models that have autocorrelated residuals, they tend to inflate type 1 errors and have greater uncertainty and so on. So by controlling that component here and just evaluating the pure non-spatial environment, the hope is that we decrease our uncertainty. But by doing so, we basically think that there is this idea that it's a spatial nuisance, and statisticians use spatial nuisance, and some ecologists as well. For many years, I've been saying that sometimes we treat space as our enemy because it needs to be tamed, controlled. But I also think that speaking about this idea that space is your friend because the spatial structure of the environments is in itself important for ecological interactions. Interesting enough, this is a very famous experiment. I always say that if there's a Wikipedia page, it's because it must be famous. So the Huffaker's mite experiment, predator-prey, 2 species of mites, one being herbivorous, the other a carnivore. And so Huffaker found that coexistence is much more possible, or at least for a few generations, when this resource itself here is more spatially less structured. There is still some structure here, but the autocorrelation of oranges. So the resource here is our origins. Those are rubber balls. And when the resource is those 4 oranges, they are arranged very close spatially, they actually go extinct, both predator and prey, because highly connected environments or predators quickly find and consume all prey, causing both populations to crash. So this is being around, this idea that spatial structure is important for coexistence, but not very much explored, at least in my reading of the literature. So I'll bring you some vignettes of the work that we've been doing in this past 10 years that sort of tries to assess the importance of spatial structure from behavior interactions in the lab up to world and global patterns of biodiversity. So this is an experiment done by Bailey Jacobson during her PhD in the lab. And Bailey manipulated food resources according to tree spatial treatments, very clumped, semi-clumped. There's a third patch here that you can't see, but they all have 9 micro patches of food. And basically, we have three food types distributions, those spatial true densities, low and high. And one of the things that we can find is that when we measure growth in the beginning and at the end of the experiment, so that difference is growth. And those are young of the year rainbow trout. So it means that within a week, actually, they can lose or gain 20% of their weight. And so what we find is that the spatial structure of the environment itself dictates the way those individuals will interact with one another. And we find that when the space is clumped and high density, there's a lot of aggressive interactions within here. And so some individuals will grow a lot, some individuals will not grow as much. But when the density is lower, then you have 1 patch per individual, and that works fine. But when you have distributed and semi-clumped, in some ways, those individuals, they move much more between. So their energy expenditure for those interactions, but at the same time, the individuals, they grow in a much more similar way. I'm not going to show the results in terms of how much they grow. They also have consequences for a phenotypic variation. So we find that in treatments where here the positive relative growth, there's individuals in patches that are very clumped and they had high variance. The individuals that grew positively, they tend to be much more better swimmers, and they have other swimming performance and capacities compared to the individuals that had negative relative growth. So my point here is that the spatial distribution and patterns of resources themselves can dictate. And in the fish literature, there's a lot of discussion because in stream fishes, in particular, we find that some populations that have a lot of variance, some fish of the same age, they're very small in the same patch, others are much bigger. In other streams, that variance is small, and it could be probably or maybe through the spatial distribution of the resources in those streams. So here's some simulation work done by Renato Enriquez Silva, also did his PhD in the lab. And so here we're interested in looking at how variance in patch connectivity. So in itself, this is a spatial question in the sense that imagine that the average connectivity among patches is the same, but some landscapes, they have a huge variance. So some patches are very well connected with others, where other patches are very isolated. So that variance in itself is capable of evolve different strategies of how different individuals will maintain themselves in a landscape. So when you have homogeneously connected landscapes, dispersal provides little competitive relief. So those individuals, they can find each other very easily. So they remain largely sedentary, and there's low regional fitness in general. And those are represented by this. So the individuals This is dispersal probability. This is local abundance. The dispersal probability is much, they start dispersing at very lower abundances when their landscapes are connected in a regular or random way. And when the variance starts increasing connectivity, then individuals start moving much later. So that also generates, this is an evolutionary metapopulation model. So it generates evolutionary strategies that are capable to resist. So individuals that evolve throughout selection in generations in more variable connected landscapes, they tend to resist those phenotypes to invasion from phenotypes that evolved in regular and random landscapes. So the spatial connectivity in itself can have consequences for evolution. So the idea here is that I'm going to move to more some of macro patterns, is that landscape ecology or the spatial distribution of features are important to meta-community dynamics and also important to biogeographic dynamics. So this is work that we started doing about 5-6 years ago into looking into another moment of spatial heterogeneity that I haven't included in those equations, but we could also include in those equations. And so basically what we have here is the spatial frequency of climates. So you can see that some climates are very common, some climates, other climates are rarer. So, and obviously, probabilistically, it's easier to disperse into more common climates than to rare climates because they're also statistically more isolated from one another. This is not necessarily a mathematical truth, but that happens quite a bit in climate across the planet at the global levels. So if a species is adapted to a common environment, it will be easier to find that common environment, that spatial replication more often, where a species that is adapted to a rare will change. And other aspects that we're looking is that temporal stochasticity is much greater in rare, not surprising, in rare environments than common environments. So there are other temporal issues going there for species dynamics. So what we find for angiosperms is that rare climate conditions, they generate limited spatial replication of environments, and they usually should be related to higher extinction and weaker rescue effects because it's just more difficult to disperse into those environments. So it filters against narrow specialists in a sense because they have to cross patches that are not very suitable to find those rare. They are usually eliminated, and we tend to find more generalist species in rare environments. So this is the standard deviation of niche climate breadth of angiosperms, and we can see that there's more variation in specialists and generalists when climates are rare. The same thing for functional diversity of those species. We find that functional diversity, because there's this mix of rarity and common or generalists, specialists and generalists, we find that actually functional diversity tends to be greater at rare climates, and that should be also, it's important for thinking in terms of conservation. This is work from Pedro Braga's PhD that analyzes the global spatial frequency of climates across the different groups of vertebrates and find more or less the same patterns that we're finding here, that there are greater variation in niche breadth when climates are rarer. And I'm going to go back to this in a few slides using simulations at the meta-community level to show that why this is the case. So the geographical or the biogeographical pattern here that we're trying to describe is that rare climates, there's few patches and low rescue. So And to cross those patches, you need to have some sort of a physiological tolerance or to be more of a generalist. And so those patches or geographic cells that tend to have to be in rare climates, they tend to have greater variation in each breath, and the opposite happens in common climates. This is work done by Louis Donnel, who used a simulation meta-community competition, competitive meta-community simulation model to demonstrate why this could happen. And so in low spatial autocorrelation, there's strong environmental mismatch among nearby sites, so it's less predictable, so there's a lot of variation in environment. So there's higher dispersal costs and risk. So it reduces effective connectivity among local communities, selecting for more generalists. And one of the things that is important in this simulation is that the same environmental variance. So here we are just changing the autocorrelation in space. We're not changing the variance. So it's exactly the same variance, but changing the where that variation is, but not how much. This has consequences for biodiversity. This is the average diversity that we find across local communities for each simulated meta community. And each dot represents replicates from the same meta community across different simulations of the same parameters. And the lines represent different dispersal rates. But the general pattern here is that as autocorrelation increases, it reduces, it weakens competitive exclusion just because species can disperse and find patches that are perhaps empty through disturbance. So there's a little bit of disturbance in those environments. And the outcome is that higher alpha diversity without reducing regional richness. This is the opposite trend for beta diversity. Sorry, this should be beta diversity, not average diversity across local communities. I just forgot to pasted the same label for the other slide. But the general idea here is that repeated colonization of similar habitats because of the high spatial autocorrelation. So the same types of species, they tend to dominate. So there's this decrease in beta diversity. So environments that are weakly structured, they tend to have greater variation in species compositions across local communities. So this is work by Gabriel Kattar and Paul Saveri, not a long time ago, where Gabriel put together a meta-community competition model. And we can see here that the increase of spatial autocorrelation is selecting for different disperser strategies. So we found three types of strategies: one in which species move, the other is species don't tend to move, and the other is that species sort of move to find more environments that fit better with their preferences. So a high spatial autocorrelation, the home body, we are calling here, Gabriel came up with his name, the home body strategy is that it doesn't pay off when it's highly structured to move much. So nearby sites are environmentally similar, but competitors are also similar and widespread. So there's a trade-off here in high local performance is best maintained by staying because living increases encounters with strong competitors without environmental gains in terms of performance. This other, when autocorrelation increases, the probability of finding species in the meta community that have the nomadic strategy, as you can see, decreases. And the context here is that environments vary sharply across space, competition is less spatially structured, and staying is risky because local conditions can deteriorate rapidly. So there is a switch from the homebody strategy where individuals don't change at higher spatial autocorrelations, spatial autocorrelation. And then to finish, there's this nomadic strategies that is selected by low to intermediate autocorrelation. Environments vary sharply across space. Competition is less spatially structured. Staying is risky because local conditions can deteriorate rapidly, and so on. So the point here is that those different dispersive strategies, they can occur and be selected in meta-community dynamics purely because the spatial autocorrelation is changing, and that's the way it mediates species interactions and how they disperse across space. This is work with Paul Savary and Jean-Philippe Lessard, in which we bring this together, this idea. As you can see, there's this interaction in how spatial heterogeneity they can select for the different types of species traits, if you will, in terms of dispersal. So the idea here is that we can take this to another level to think that there is variation in landscape structure in terms of the spatial structure of the environment, autocorrelation, or heterogeneity. But the species pools also, they may vary. In some, you could have species that are very similar to one another, and you could have other pools that the species are very different from one another. And depending on the type of heterogeneity and spatial autocorrelation, you're going to end up with different dynamics. And those are sources of contingency in which you expect different patterns of species distributions just because the way the spatial heterogeneity in itself varies. So at the end, there is this interaction between variation in means and spatial autocorrelation at different levels. And I think what we need to start doing is more measuring this sort of patterns and trying to understand what those different components could mediate interactions. And so those different spatial dispersal capacities that vary across different pools and the heterogeneity, they can dictate different patterns of alpha and beta diversity. And we explore this again in this paper. So it's just this recurrent theme in which we see that the way the spatial structure of dispersal networks, they will dictate dispersal rates, but also depending on the types of the interaction that you find between species traits or species often dispersal capacities, this will change. So for this combination here, sites are highly connected here as well, but there's more variation in trait among species here than here, and the same. So there's this interaction between the spatial structure of landscapes and the traits that will dictate through species interactions and dispersal patterns, how different beta diversity and alpha diversity. So I tend to say that I tend to work in this forefront of the Massey community ecology, going back to John Lawton's sort of thinking that community ecology is Massey. There are others that came up with similar ideas since then. But I think that one way to start thinking about contingency is to dissect those spatial components of mean variation, variance variation, autocorrelated variation, and to different places from local, from small regions to global, those components will change. And I think that we need to understand how they change, and perhaps we could have more synthesis in terms of this contingencies that are happening. This work couldn't be done without a lot of people throughout ideas and collaborators and some sort of funding. So with that, I will finish for now. Thank you again.
Thank you very much, Pedro. That was really, really great, really interesting also, and just comes to show how complicated ecology and evolution can be. Okay, well, I will invite everyone to please put your questions in the Q&A box and I can read them off to Dr. Peres-Neto. For the person that asked about the presentation and the video session recording, we will post that video on our YouTube channel, so you can find it within days of the seminar. So yes, it will be available to everyone to take a look at and listen to again. Okay, so as the questions are rolling in, I can sort of, I was going to kick off with the first question, but there's plenty of questions to ask already in the Q&A box. So let me read off the first one from Brittany Pooh. This was a really interesting talk. Thank you. I am interested in how you characterize human factors in your models, as these are extremely key, but often hard to get data for. And human processes operate at different scales, temporally and spatially.
Yeah, that's an excellent question. So in the systems that I work most comfortably, fisheries and lakes in Canada, we have incorporated, if you want to write to me, I could send you a couple of references. One of the ways that in fisheries, so we can have actually very good assessment in terms of human impact on fisheries. There's a lot of good data in Canada, particularly because of when we go out fishing, we need to purchase licenses in most cases, and there is a track, and there are measures of human impact and so on that are done. And we find in general that although it's not a very strong pattern, but a lot of the lakes that we work, it's just such a large scale that the impact might not be big enough. But certainly one of the things to start looking, and I think landscape ecologists have done that, is to look at how human impact or human footprint or the footprint of their impacts, they could change the way spatial structure is organized. You're probably going to notice that a lot of the themes, that a lot of the data that we're using is climate. And so there certainly would be, we know, we've looked in the past how climate is changing. Obviously, the average is increasing for temperature and other aspects, but also the spatial structure and the variance is changing. Very little work has been done on that. And we have very good models and we have excellent data at the global level to actually characterize climatic variation across those different components. So this is work that we've been looking right now, is to decompose these levels of variation that we find and perhaps have a better understanding of how humans are impacting climate as well for changing heterogeneity and spatial structure, which will then affect, obviously, biodiversity dynamics. Thanks for the question. It's a really important one. Is often I don't cover enough in my talks.
Thanks, Pedro. I'm just gonna keep on going with questions because there's quite a few in there. And the next question, there's actually 3 questions from Stenio Förster. I will just start with one and we'll go one by one so that we don't miss anything. Could you rank the 3 most important aspects that prevent us from linking pure spatial effects to neutral-based processes and pure environmental effects to species sorting or local adaptation when explaining variation in species composition?
So can you repeat again?
Yes.
The first part.
Could you rank the 3 most important aspects that prevent us from linking pure spatial effects to neutral processes and pure environmental effects to species sorting or local adaptation?
So I would rank 3. It's a really interesting question when you put someone against the wall and you have to think about this. One of them are the statistical effects. So we explore Sylvie Clapp, who we work together in. We know that the spatial signature of environments can be confounded with neutral dynamics. And so that's one challenge, is to separate that statistical expectation that exists. We explore this in a paper. If you're interested, you can send me an email. It's called Beyond Neutrality. It's a paper published in Ecology a few years ago. So beyond neutrality is this idea of how to actually separate the elements that could be neutral from the elements that are expected in terms of statistical expectations from autocorrelated environments. The second one is that in a lot of the models that we looked empirically, space can represent many things. Space is not, it's very hard to actually, that's the third challenge, but it's very hard to incorporate dispersal dynamics into those large scale models. The reason is that many environmental features that may not have been measured, they can also have a spatial structure. You could have also species interactions that can lead to spatial signatures, but you did not incorporate into your models. So by having that spatial signature could mean many things. And there's some work that discussed this quite a bit from others. I would be happy to send some of that, but the literature is somewhat rich in debating. And measuring dispersal, we can make assumptions. We rarely ecologists that look, there has been a lot of work using variation partitioning, for instance, to dissect neutral from species sorting dynamics. And a lot of them don't incorporate species dynamics or species dispersal. You can make certain assumptions and you could actually incorporate that species dispersal dynamics into those models. There's very little work done that. There's lots of work in meta populations, but there's very few work done in meta community ecology. So those are the three for me, is this the spatial expectation between two sources of variation, neutral and species sorting. The second one would be the spatial component that we represent statistically could mean many things. And the third one is assess dispersal in itself. It's not easy.
Okay. Well, one more question from this person, from Stenio. To what extent do spatial scale and sampling design, regular versus irregular, influence our conclusions about the contribution of the pure spatial component to explaining compositional variation? Would it be an overstatement to say that spatial structure is always important at the broadest geographic scales?
Yeah, that's certainly true. And perhaps I enjoy working with lakes because they are more or less natural patches, and the design is more of a natural. However, we don't always sample all lakes, so there will be aspects of design in itself. There is actually very good work, including from Canadian researchers, that looked into how the design, the spatial design, sampling design in itself can influence patterns, and definitely. The answer is not easy how to do it, but we know that that can affect. So I think that there are ways in which we could think about how to remove or potentially reduce uncertainty related to sampling design. Is not a place where I particularly spend a lot of time. I should. But some do, but it needs to be done more carefully, because we know that at macro patterns, a lot of the design can certainly drive. We also know that not only design, even if we work with cells, that we change scales sometimes. If you use a 10 by 10 kilometer, 3 by 3 kilometer, 50 by 50 kilometer, even if you use complete sampling, just the way you actually impose that will change the scaling in which different processes are happening. Now, ecologists tend to think that because you're scaling this way or that way, we're finding more or less what processes is driving ecological organization. There are also some assumptions in there that we don't think too much, but they can complicate things, that they can also generate in some ways, patterns that are artificial rather than true underlying the true ecological organization or structure that we're interested in.
Thank you, Pedro. I'm certainly guilty of looking at scale dependence and maybe not like making inferences that perhaps we should all think a little bit more deeply about. Stenio, if that's okay with you, I will move on to the next person's question, just so that we can get as many people's questions answered as possible. So the next question is from Mark Lomolino. Species traits such as niche breadth and dispersal capacity are strongly correlated. To what degree does spatial structure and covariation influence covariation of species traits?
Oh, that's thanks, Mark. That's a great question. Certainly true. Although we don't find that all. It depends on the dimension of niche breadth that we're considering. So one of the things that we didn't present this, and some work that we're thinking how to assess that is whether, for instance, is there a trade-off that could exist that the way the types, let's say climate or environments that you prefer, that you have a greater affinity, those might actually not correlate, for instance, with the climates that you have to cross for dispersal. So you could break maybe that correlation in some aspects or very complex aspects, and those are things that we've been thinking. It's not clear to me yet how this work will look like. I would love that others start thinking about this, but my general thinking is that there should be, if species are really well adapted to their landscape, it should be that the niche breadth, which is in itself multidimensional, right? We consider this volume, but in itself, different, for instance, different types of environments will contribute more or less to that ability to sustain and disperse for those environments. So the short answer is I don't know. It's a great question, but I invite others to look at that. I'll certainly continue to think more about this.
Thank you, Pedro. Ramesh Kataria is presenting an example from his own work and simply asking whether he can reach out to you via email to get some advice on his research.
By all means, I'm always happy to jump on a Zoom call. Sometimes it's more helpful. I would love to do that. Thank you.
Yeah. Okay. So Ramesh, you can probably find Pedro's email online when you just Google Pedro's name.
If you type Pedro Concordia, you'll find me.
Excellent. An anonymous person is asking, you mentioned climate commonality and the generalist specialist continuum. Can we disentangle this from resource availability, or is spatial aggregation already considering resource distribution?
Well, that's a complicated question. Sometimes I work on the lab because I can manipulate resources. I haven't done a lot of work with resources themselves. I tend to think more about environmental variations such as climate and so on. So I'm kind of I think they definitely there must be some level of interaction where there is often this trade-off between resource availability and preferred environments. It's not clear to me. I'll probably start thinking for an hour after the seminar how the spatial heterogeneity and covariation could actually modulate potential trade-offs between those. One of the things that you probably noticed, I didn't expose that too much, but I tend to bring languages of trade-offs that I try to think, especially in the modeling work that we do, so that we have a better understanding of those different aspects in terms of selection of dispersal strategies or phenotypes and so on. So I'll continue thinking, but nothing on top of my head. It will depend on how the resort. So yes, I had an undergrad student that now I remember that looked to a model in which we did a little bit of game theory to look at the spatial distribution of resources and the spatial distribution of environmental variation. We end up not exploring that further, but now you made me think that I have to go back to that work after 15, 16 years.
It's great when questions prompt us to go back and look at other questions in more detail. I am really sorry we are out of time and Pedro needs to teach in a little bit, so we really cannot stay much longer. There were many more questions in the Q&A, but we just cannot get to those today. I assume that Dr. Peres-Neto would be okay with you reaching out to him via email if you have questions that need to be answered. Otherwise, please come to our next seminar in February. Thank you very much, Pedro, for coming. It was a fantastic talk, great attendance, great questions, and thank you to everyone who came. And I hope to see you all in February, just a month away from now. Thank you, and till next time.
Bye. Thank you,
Everyone.
everyone. Thanks, Hikaru, Martha.